dog, man.
Okay, I think we're ready to roll here. So we are going to finish chapter 30, which is seedless plants. So we'll talk about the Lycophytes and the ferns and their allies. And then we'll start chapter 31, which is the seed plants, and get into gymnosperms and angiosperms. So Lycophytes is a sister group to all of the vascular plants. Remember, we discussed bryophytes. They don't have true roots, leaves, or stems, no vessel elements, if you will. So we're going to start talking about vessel elements and all that stuff. So similarities to vascular plants with the Lycophytes: their leaves are small and they are unbranched. They just have that parallel vein. Where when we get into more complex vascular plants, you see that their leaf size increases, and we see complex branching of the veins inside of that organ. I'm going to show you a picture of a Lycophyte, and you're going to think that it's a moss.
Her name used to be Singleton.
Okay. Thank you.
I don't know if that's what it is now, but that's what it used to be.
Singleton. Okay.
I knew it'd come to me.
For reference, I asked Sue from Breckenridge what's her last name, so that's because I don't know her last name. All right. So the Lycophytes today are really, really small, but in ancient past, due to fossil records, they were huge. They were trees. But we can find Lycophytes worldwide. They are most abundant in tropics because of that moist environment. And they now today, because they're small, you would think of them as mosses. So that's why they're called club mosses as well. We still have spores. We don't have seeds. We have spores that will be released, and they germinate, forming that gametophyte, and the gametophyte will produce the egg and sperm and then turn into a sporophyte. Okay, so here's a picture of Lycophyte, club moss. Okay, so we still don't have true roots. We have a new thing called a rhizome, which is a modified stem. But if you look at it, you're like, oh, it kind of looks like a moss. Okay, Lycophytes also have this structure called the strobili. And I'm surprised your book really doesn't mention it, because in the old edition they were all about the strobili. But yeah, I really couldn't find it in reference. So these are the fossil Lycophytes. Okay, they were very tall, tree-looking like structures. Okay. Here's the life cycle for a club moss. It's very similar, but just note that they are homosporous, so they only produce one spore, which means that from that spore, both male and female gametophytes can be produced. Okay. So let's just start with fertilization. Egg and sperm meet. We form a zygote, an embryo to the sporophyte. And then on the sporophyte, we have a structure called the sporangia. And inside the sporangia are the spore mother cells that go through meiosis to form the spores. Spores get released. They germinate into that gametophyte. And the gametophyte, through the process of mitosis, forms the sperm and the egg, the antheridium and the archegonium. So just a generalized life cycle. Here's what it actually kind of looks like. With fertilization, egg and sperm meet. There's the gametophyte. I know it says root. They really don't have a true root system. There's something called a rhizome, and I think I talk about that in the next section. But inside the strobulus, you find the spore mother cells that are released, and those spores get released, forming that gametophyte. All right, select the plant that is seedless and vascular. It is D, Lycophytes. Yeah, that's it. Yeah. Okay, moving on to Pterophytes, ferns and their relatives. So I'll talk about some features that they have and then contrast them to a pterophyte and the moss sporophytes. Okay. So Lycophytes, bryophytes, and pterophytes all have antheridia and archegonia, so structures that form the female haploid cell and the male haploid cell. They still are kind of tied to water for the process of fertilization because their sperm are flaginated. So they are still tied to water, whereas when we talk about seed plants today, they are non-flagellated sperm. So one group of the fern and its relatives is called the Whisker Fern, or Coelophytes. Whisker ferns still don't have true roots, and they also don't have any true leaves. And through the course of evolution, it looks like they lost them secondarily for whatever reason. They occur in the subtropics and tropics, so again, warm areas. Unique feature about whisker ferns is this dichotomous Y branching. And you know, you can understand why they call it a whisker fern, because you turn it upside down, it looks like you could stir something with it. Their gametophytes are pretty much colorless, so again, small, microscopic almost. And because they don't have true roots, they tend to form symbiotic associations with fungi to help them absorb nutrients as well as water. All right, another relative of the fern, horsetails, and this is probably one of my favorite plants out there.
Those things are so hard to walk through.
Oh. Well, when I see them, I'm hiking on a trail and they're on the boundary, you know, and my daughter likes to pull them because they look like a straw, you know. So she'll just like grab it from the root and then she just pulls them apart, keeps her occupied. Yep.
I was walking, like hunting, and I got stuck in them. Like they were so thick I couldn't move. And I was yelling for my dad because they were like 6 foot tall, and I can't see. So I'm like, Dad, I don't know where you are, but I can't find you.
So they do have these jointed stems, and yes, you can pull them apart. Okay. They again don't have true roots. They have something called rhizomes. So rhizomes are stems. They are photosynthetic. They run horizontal to the ground and then they kind of sprout up. You can see that they are jointed and ribbed, and they actually deposit silica, okay, so sand. So during the 1800s, the Great American West, they actually would use horsetails to like clean their cookware because it was like a scrubber, a natural scrubber. So scoring rushes. In today's terms, I think there's only about 15 species of horsetails left. But during the Carboniferous period, these guys could get tall, like 15 meters. So like 45 feet, could you imagine? Yeah. And so I guess one more thing I want to talk about. So when you look down, this is a picture I took. When you look down, you can see it, it's hollow, and that's because a lot of times you find these in wet, low oxygenated areas. So this is how they take in CO2 and release oxygen. So some more pictures of horsetail. This is obviously a fossil of horsetails, and then today's horsetails at the top. You can see that they're jointed and ribbed, and you can actually see some silica deposits going on there. When you zoom in microscopically, this is where the spores are found, kind of underneath that strobulus structure housed in there. So then when it dries out, then they kind of flap out and fly. All right, the final group underneath the Tropophytes here are the ferns. So they are the closest relative to seed plants. I was kind of going it from ancient to, more common recent Tropophytes. So they are very abundant. They, again, don't have true roots. They have horizontal rhizomes, and from them they can grow very large leaves, and you'll see that on the next couple slides here. The leaves, we don't call them leaves, we call them fronds, and they're usually compound, which means they kind of match on either side. So, yeah. They can be divided into many leaflets, and on the underside of the leaves, or the fronds, you find these clusters, little dots, if you will, and those are the sporangia, called sori. and that is where the spores get released. Okay, so this is kind of what ferns look like in nature. They actually can coil up, and these are called fiddleheads. I think it's just so cool. Like, and there's the rhizome and where it branches out, but it rolls up, and obviously they call it a fiddlehead because it looks like a part of a fiddle. But then it can uncoil and then show you the compound leaves. So there's the compound leaves going on here, and then on the other side you have the sori. So these are sories that release the spores. Okay. All right, I'm going to show you a video here. So sometimes they have spring-like devices called, well, it just looks like a catapult. So I'm going to show you a video, but how it works is inside we have the spores and we have this outer covering, and these are the lip cells. And when it gets dry or it desiccates, then it kind of cracks open, and then due to osmotic pressures, you're going to see in this video, it like just springs back and then just throws these spores. So it's like literally a nature's catapult. So click on this link. Hopefully the internet is working. And hook up my speakers. Thanks.
The movement of plants is nowhere more obvious than in the infinitely diverse mechanisms used to eject the spores. In the large group of ferns known as the leptosporangiate ferns, spores are ejected by a mechanism that is aptly described as a cavitation catapult. The spores are kept in sporangia and in small clusters, the sori, under the fern leaf. Close examination of the sporangia, when exposed to air, reveals a slow opening of the spherical capsule, followed a rapid closure during which the spores are ejected. To understand how the spores are ejected, we must look more closely at the structure of the sporangium. The sporangium is a thin spherical capsule that encloses the spores. A row of specialized cells sits to one side of the sporangium. The cells of the annulus have thickened inner and radial walls, forming a reinforced U-shaped structure, while the lateral walls are rather thin. When the cell loses water to the environment, its volume must decrease. This is accomplished by bringing the radial walls closer to each other and by collapsing the lateral walls inward. This clip shows the intricate relation between the decreasing cell volume and the curving of the annulus. Over a period of a few seconds, the annulus reverses nearly completely its curvature in response to the ever declining cellular volumes due to evaporation. The strong curvature that develops along the annulus must be supported by balancing force. This force is provided by the suction force, or negative pressure, that the cell content exerts on the cell walls. The continually decreasing cellular volume and bending of the walls invariably led to more negative pressures within the cells of the annulus. Beyond a certain point, the pressure within the annulus cells is so high that their content cavitates. Various estimates indicate that a negative pressure of 100 to 200 atmosphere is present within the cell at the time of cavitation. Cavitation effectively releases the suction force that was holding the annulus in its bent configuration. The annulus thus returns quickly to its original configuration, ejecting the spores in the process after this nucleation of bubbles. However, The greatest refinement comes into play only after cavitation. The spores would not be ejected if the sporangium simply sped up to its closed geometry. Its returning motion must be arrested abruptly for the spores to be ejected. The same problem was faced by the designers of the first catapults. In catapults, a crossbar stops the arm in its course, thus allowing the projectile to be launched. This crossbar is strikingly missing in the Fern sporangium. We have found that the sponge-like organization of the annulus walls allows for the two very different closing timescales that serve the same purpose. The fast timescale is inertial and is observed in any elastic material that is suddenly released from a different geometry. The second timescale comes from the need for water to flow across the annulus wall to adapt to the rapidly changing geometry. The flow of water within the tiny pores of the wall is impeded by viscosity, leading to an abrupt arrest of the sporangium motion. It is remarkable that a dozen cells have evolved to fulfill all the critical functions of a catapult.
Okay. So yeah, not human. I was gonna say biology and physics. Who knew? All right, so the life cycle of ferns. We'll just kind of go through it here. Let's start with fertilization. Okay, so egg, sperm. Sperm's gotta usually swim to it. We have a zygote that is diploid. It turns into our sporophytes. You can see that we don't really have a true root system. We just have rhizoids. Oh my gosh, my phone's going crazy. But from here, we form our large sporophyte. Our sporophyte is larger than our gametophytes. You can see young leaf fiddleheads down below from the rhizomes. But if we zoom in on the sori, okay, you can see this immature sporangium. That's the catapult that you just saw in that video, where it will go through meiosis, produce those spores. Spores get released, and the spores will germinate into a young gametophyte that is smaller than the sporophyte. Okay, so yeah. So that's a gametophyte. It's the sori. Okay. All right. So we started this chapter talking about the chlorophytes to the charophytes. These guys are the ones that gave rise to land plants. We discussed the bryophytes, where we have cuticle and antheridia and archegonia. The hornworts have a stomata on the end of their tapered sporophyte here. Discussed the lycophytes because they have a vascular tissue, stems, roots, leaves, some have specialized structures for that. So now we're going to transition into gymnosperms and angiosperms, where we have the development of seeds and fruits and flowers. So review question: which of the following sporophytes attached to gametophytes and nutritionally are dependent upon them? I think I worded that weird, but which ones have sporophytes that are attached to their gametophytes and are dependent on the gametophytes for nutrition? It is E. Okay, so I threw this question because I was like, oh, let's just see if they remember bryophytes. Okay, so liverworts, mosses, and hornworts, they are all bryophytes. The sporophyte grows. It's parasitic to the gametophyte. It needs nutrition from the gametophyte. Next question: what is the proper name for the horizontal stems of ferns? C. Rhizomes. Okay, rhizomes. Name the plant that has neither roots nor leaves. It is B, Whisker Fern, because it lost them secondarily. Okay.